Hey Bio30s, Manchak here with your last video lesson for Unit C. And we are going to cover concepts C10 and C11. But before we get into that, I want to work our way through what we've done in the unit so far. So we started out talking about cell division in the first three concepts. We talked about mitosis and meiosis. Then we moved on and we started to talk about a little bit about non-disjunction and what goes on when mitosis or meiosis doesn't occur properly. Then we jumped into what we called classical genetics. So we started to talk about Mendel and his laws of dominance, segregation, independent assortment. Then we got into patterns of inheritance and discussed a whole bunch of different ways that we can determine probabilities of whether an individual might look this way or that way, depending on what the parents look like. We went into and started to talk a little bit about the research of Morgan and other patterns of inheritance that came out from that, like sex-linked characteristics. We talked about gene linkage and crossing over and how we can relate those. Then we did a bit of a history lesson in C8, and we talked about what went on to discover that it's actually DNA that was the molecule for inheritance. Then lastly, what we moved on to in the last class was talking about the DNA molecule itself. And we had a good close look at its structure, including the sugars and the sugar phosphate backbone and the nitrogen bases that go down the middle. And we talked a little bit about the process of DNA replication. Where we're moving on to today is that we're moving into concepts C10 and C11. Like I said, the last two concepts in the unit. These are going to be relatively lengthy concepts. So what I've decided to do is split this up into two videos. So this is video 1 of 2 for this week. And then the second one is going to cover concept C11, meaning that this first one will cover concept C10. And you can break it up a little bit. I have a feeling these are going to be hefty concepts, so I'm going to skim over some things and let some video links that I have for you do some explanation as well, so you guys don't have to listen to me the entire time. So concept C10 is all about protein synthesis. It's all about taking what's in the DNA molecule and translating that eventually into a protein molecule, which gives us the appearance that we see for whatever gene that we're trying to express. Okay, so we're going to talk about things like messenger RNA, transcription, translation, amino acids, all of those kinds of things. So protein synthesis is really what C10 is all about. C11 is going to jump into talking about, okay, we know all of this stuff. What do we do with it? How do we work our way through that? How can we use it? So we're going to talk about things like recombinant DNA. Transformation, taking DNA from one creature and sticking it into another creature and making that work for us in some way. Then we're going to talk about mutation and the different kinds of mutation and what the evidence is out there for the mutation stuff. All of this is going to lead us to transition into Unit D next week. And Unit D is going to take some of our genetic stuff, but we're going to look on it on a grander scale. Rather than looking at individuals or even just the DNA molecule itself, we're going to look at ecosystems and much broader contexts with some of this genetic information in it. So I'm going to slide myself over here, and we'll just do a quick run through of what we're going to cover in C10, and then I'll run through this again in the next video for C11. So it's going to be a long one, which is what I said. This is going to be, I think, a long set of videos. So be prepared to make sure that you guys have the time to run through the video and stay on top of things. In C10, we're covering gene expression or protein synthesis. So I'm going to talk to you guys about transcription and translation. Basically, how do we get from a DNA molecule to a protein? That's what we're going to cover in C10. I set aside a couple of videos for you to have a look at, and these will be in the Google Classroom under resources for C10 and C11. So find these videos in there. The one on protein synthesis from Amoeba Sisters, we've used them all the way through this unit so far. Go back and have a look at this one again, please. And then this next one is sort of a continuation of ones that we've seen before. In fact, the middle sequence on DNA replication is a video that I showed you in the last lesson. But this one ties everything together. It's a look at the DNA molecule, then DNA replication and what that looks like with big Okazaki fragments coming out, all that kind of stuff, and then the process of transcription and translation. You guys will have some practice questions out of the textbook, as well as a worksheet to work on for C10, which the key is up on the Google Classroom as well. And then once you're done the C11 stuff, you can move into doing the practice quiz and then ultimately the summative quiz after you've received your feedback. So I think that's enough introduction. Let's actually get into talking about or talking about this last concept here. So I'm going to readjust where I'm positioned here and maybe slide this window out of the way so you guys don't need to see it anymore. Boop. Okay, so gene expression is where we're going on into. So we're going to describe how genetic information is contained in the DNA molecule, and we already know that, but how do we get from that DNA molecule to an actual assembled protein? And so we're going to play around with that a little bit and see how that works for you. So we already know that genes are basically specific segments of DNA that code for a particular protein. Okay, so a chunk of DNA on a particular chromosome is a gene, and that gene codes for a protein. Now, we saw in the previous sections, in the previous section of this unit, that those genes can be broken up into alleles, which are just slightly different physical expressions. Maybe it's the difference between, dark hair versus light hair, or whatever it might be. But that is still coding for the color of hair, which is the gene that we're trying to express. So gene expression is the conversion of a gene into a specific trait through the production of a particular polypeptide or protein. Okay. Remember from Bio 20, a polypeptide is many peptides stuck together. And let's keep it simple. A peptide is 2 or more amino acids stuck together. So a polypeptide is a super long chain of amino acids all stuck together. And then the trick I want you guys to remember from Bio 20 is a protein isn't a protein until it's bent, folded, and twisted into a particular shape that gives that protein the function that it might be. And we know from our explorations last year that a protein might have several polypeptides in it that are manufactured independently of each other, and then they come together and make that particular shape that's bent, folded, and twisted into the particular structure that we want. Polypeptides, remember, are chains of amino acids, and we said last year amino acids are the building blocks for proteins. So where do we head next? How do we make a protein? Making a protein involves 2 processes, okay? These processes are known as, and I'll get out my pointer here, these processes, that wasn't it, are known as the central dogma of molecular genetics. Molecular genetics being the stuff that's really happening on a molecular level. Central dogma means this is the stuff that has to happen all the time in order for us to make proteins, okay? And this happens in every organism all around the world because we all have DNA and we all have proteins, and we need a way to manufacture that DNA. Oh, got an itchy nose. So making a protein involves transcription and translation, okay? And it's actually really remarkably elegant how this works. We have to first read the DNA molecule. The problem is the DNA molecule is locked up inside the nucleus of a cell. Because it's locked up inside the nucleus of a cell, we need to find a way to get the DNA, the message, basically the information that we have in the nucleus, out into the cytoplasm of a cell. Remember, this is only true for eukaryotic organisms that have a nucleus, but for now, we're just going to go with this, okay? So we've got this DNA inside the cell. We want to make a particular protein. So what we have to do is we have to transcribe, this is transcription, transcribe that section of DNA, that gene, onto a little messenger piece, and we'll talk about what that messenger piece is in a second, and then we're going to send that out into the cytoplasm. Once it's in the cytoplasm, it is going to get translated by a ribosome, which we talked about way back in Science 10. So it's going to get translated by a ribosome, and that ribosome is going to simply read that message, read that gene on that little messenger piece, and eventually piece together a protein, a sequence of amino acids into a protein. Okay, that's basically transcription and translation. Transcription, remember, takes place inside the nucleus. Translation takes place inside the cytoplasm, and we'll go over that again in a second. Each stage, each thing, transcription and translation, each involves initiation, elongation, and termination. And we're going to talk about what those are in a second. What we haven't talked about yet is ribonucleic acid. which plays a very important role in what we're talking about with the making of a protein. Ribonucleic acid, I think I alluded to it back in the last lesson, is very similar to DNA, except that it's got three things that are different about it. So pay attention, and please know these because I will likely ask about them at some point. First off, a ribonucleic acid has an extra hydroxyl group on the sugar. I think I mentioned that in the last lesson, meaning that the sugar is a ribose sugar, not a deoxyribose sugar. So this is why we call it ribonucleic acid, because it's got a ribose sugar, as opposed to deoxyribonucleic acid. The other thing is that RNA does not have thymine on it. Instead, it has something called uracil, also represented as U. So in any place where you would find a thymine molecule on RNA, that has been replaced by uracil, okay? And I'll get into how that works when we get into the actual transcription and translation bit. RNA is also single-stranded. DNA is double-stranded. Remember, it's twisted in that double helix. RNA is just a single strand floating around, moving from place to place as we need it. Okay, so those three differences I'd like you to know. So let's jump into transcription. What is transcription? How does it work? Transcription involves basically genetic information being transcribed or transferred from DNA to messenger RNA. Okay, messenger RNA is a specific kind of RNA. It's still RNA, but we have this little lowercase m in front of it, mRNA. M referring to messenger. Okay, messenger RNA carries genetic information from the chromosome, which is inside the nucleus of the cell, to the site of protein synthesis, which in eukaryotic cells is going to be the cytoplasm. Okay, if you're in a prokaryotic cell like a bacterium, you're still going to have that messenger RNA. It's just that the DNA is free floating. There's no nucleus, but we're not going to get too hung up on that right now. So transcription is the process of taking that DNA molecule, reading the gene that we want, okay, and then transcribing it into messenger RNA. And then that messenger RNA snakes its way out of the nucleus, carrying the genetic code for that particular gene, and finds something in the cytoplasm to translate that little piece of messenger RNA into a protein. So let's jump forward here. So here's how this works. Remember, I said there's going to be three parts to transcription and translation: initiation, elongation, and termination. So here's the initiation portion. An enzyme, to start this whole process, an enzyme called RNA polymerase, binds to the DNA at a specific site, what we call a promoter region. A promoter region is just simply a string of adenine and thymine bases in a nonsense pattern, just there to signify, hey, yo, look over here. This is the beginning of a gene. Okay, so it's a sequence of adenine and thymine bases of nonsense meaning it doesn't have any genetic code to it. It's just a bunch of A's and T's signaling the site at the beginning. That's where a beginning of a gene, that's where the RNA polymerase grabs onto the DNA molecule. So, and you see, if we see in our diagram here, I'm going to move myself out of the way, we have an RNA polymerase enzyme that's going to find a promoter region and grab onto it. So it'll grab on over here some way, right? Where our nonsense is, and then we've got our gene that it's going to try to code. Okay, so that promoter region just indicates this is where you need to start reading. Okay, and then that RNA polymerase is going to move down the molecule, which we'll see in a second here. So I can get it to click. So the RNA polymerase uses one strand of the DNA, what we call the template strand, to build the messenger RNA. And simply all it does is it unzips that double-stranded DNA, sort of inserts itself in and runs itself along that molecule, okay? And it builds in a 5' to 3' direction on the RNA molecule, but it reads in a 3' to 5' direction on the DNA molecule. Okay, and so we don't transcribe the promoter, it just uses that to orient itself. Then it builds its mRNA strand, and that's this blue strand that you can see at the bottom here. And it runs along until it gets to the end of the molecule. So right now we are in the elongation stage. initiation was finding that promoter region. Elongation is unzipping that DNA molecule and then running yourself along there. Instead of using DNA, you're using messenger or RNA molecules, little RNA nucleotides that are being added in a complementary fashion. So if there is a T, it's going to add an A RNA nucleotide. If there is a C, it'll add a G. If there's a G, it'll add a C. If there is an A, it will add a U. Okay, so every time there should have been a thymine, it's going to replace it with a uracil molecule. Okay, hope that makes sense. So that's elongation. Then the last step here is termination. Once it reaches the end of a gene, it recognizes the end of the gene, and when it comes to what's called a termination sequence. And I'm not going to jump into that too deeply at this point, but once it gets to the end of the gene, it stops. The RNA polymerase disconnects. The RNA itself, the messenger RNA that's newly been synthesized, gets disconnected from the DNA molecule. The DNA molecule zips itself back up, and that messenger RNA floats its way out of the nucleus and into the cytoplasm to be translated again. At this point, I'm not going to get you guys to watch any videos just yet, but let's just summarize that really, really quickly. So that's the transcription process, which takes us from DNA to messenger RNA. That messenger RNA, when we're done, carries the code that a ribosome needs to translate into a sequence of amino acids which manufactures a protein. There was 3 steps. We had the RNA polymerase attach to a promoter region and find the beginning of that gene. Then it elongated. It ran down that gene by unzipping the molecule and running along and building a sequence of messenger RNA that is complementary to the template strand that it was reading. When it got to the end of that gene, it terminated, disconnected, mRNA disconnects, DNA molecule zips back up. Okay? Hope that makes sense and that's not too confusing for you. I did a lot of arm waving. I tend to do that for some reason. I've noticed this on my videos, so I hope you enjoy that. It seems like every preview that I have on YouTube is me going something like this, or I'm moving my arms and doing weird things. So, hey, do I do that in class? I wonder. Anyway, this is another just quick diagram in your notes there of what it looks like. In this case, we're reading the top one here and building that sequence. And what I'm showing you here is that we're building that complementary strand of messenger RNA. There's our RNA transcript right here. So if there was a C on the strand, then the RNA polymerase plunked in a G. If there was a T, it plunked in an A. If there was a G, it plunked in a C. If there was an A where it should have been a T, it's put in a U instead. Okay, that's what transcription's all about. So if you wanted to pause right now, there's a couple of questions on page 669 you could do just to reinforce this stuff, and hit pause now if you want to. Otherwise, we're going to continue on to translation. So to set the stage, we've got that sequence of DNA has been read and transcribed into a piece of messenger RNA that has snaked its way out of the nucleus and into the cytoplasm. Now what needs to happen is we need to take that and turn it into a protein. We need to translate it into a protein. So the genetic information carried by the messenger RNA leaves the nucleus and is used to synthesize a polypeptide chain, a protein. The polypeptides are assembled by sticking together amino acids. Okay, remember there's 20 possible amino acids, and I'm going to pull something up in a second here to show you, to help you with a little bit of what's going on here. During translation, the DNA is read in groups of 3. Not the DNA code, pardon me. The DNA code is in the messenger RNA. And so what happens is when it finds the ribosome starts to read that messenger RNA sequence in 3 nucleotide segments, what we call codons. Okay? And each codon codes for a specific amino acid to be placed on a growing polypeptide chain. Just a couple of things to note. There is what we call a start codon. The start codon on the messenger RNA is AUG. That also codes for methionine, which is one of the amino acids. So every gene that we are going to code starts with a methionine amino acid on the end of it, okay? Or on the beginning of it, pardon me. Then each of these sequences also comes across, each of these messenger RNA sequences it will come across a stop codon, and there are 3 stop codons, what we call terminator codons. And when it comes along to that stop codon, boom, we know that we've hit the end of the gene. That's where the end of the amino acid sequence is. That's the end of our protein at that point. Now, this is where I'm going to bring in, hopefully relatively easily, this right here, which shows you, now this is your data booklet, and your data booklet is in the Google Classroom right up at the top of the classwork page under general information. It's got a bunch of stuff in it. I believe I mentioned it to you in concept C5 when we did some pedigree stuff, which is right at the end of this page here. What I want you guys to note right now is there is this page, which is messenger RNA codons and their corresponding amino acids. Now, I'm just going to zoom this in a little bit for you and show you a couple of things, just so it's a little bit bigger. Well, we've got this up. There we go. That looks perfect. So this is messenger RNA. Often you'll be given a piece of DNA, and you'll have to transcribe it into messenger RNA, and then you'll read that messenger RNA in 3 nucleotide segments to make up your amino acids. And so you can see all the amino acids listed on here. Okay, I'm going to go over here to methionine to start. AUG is an initiator codon, and it also codes for the amino acid methionine. So at the beginning of a gene, you will always find AUG, which you could write down as start or methionine, or both if you'd like to. And then you're going to fill in a bunch of amino acids. So if your messenger RNA says CCC, that is a proline amino acid. If your messenger RNA says AAG, that is a lysine amino acid. I'm going to point out those 2 here plus this one over here. Those 3 are your stop codons. So your ribosome is going to read along that messenger RNA sequence, and if it comes across a UAA, a UAG, or a UGA, it knows at that point that is where the protein stops. It will stop reading at that point, and now it's assembled that chain of amino acids that it needed to make that particular protein. Okay, I'm going to keep this handy and drag it off the screen for now, and I'll bring it back up in a second here when we do some practice. Okay. So here we go. This is what I just brought up for you, so I'm going to skip over that right now, but you can find that in your data book. It might be handy to have it with you as we move through the examples in a minute here. So translation begins when a ribosome, so here's our initiation again, but the ribosome binds to a messenger RNA at the start codon, and sometimes it has to move back and forth until it finds that start codon. The ribosome is made up of a large unit and a small unit. So there's a large unit on top and a small unit on the bottom. You guys will see what that means in just a second. The 2 subunits sandwich the messenger RNA molecule and start to read it. The ribosome moves along the messenger RNA strand, making a new amino acid every codon. Remembering a codon is 3 nucleotides, so an A, G, G, or something along those lines. Okay. But how do the amino acids get to the ribosome? Now, this is where we're going to introduce you to another kind of RNA called transfer RNA. Now we're moving into elongation. So floating around in the cytoplasm are little transfer RNA molecules, tRNA, transfer RNA. And what they do is they float around in the cytoplasm and they pick up specific amino acids. Okay, and they have one end is a piece that will fit as an anticodon to the mRNA molecule. The other end has a special attachment point for an amino acid. So they'll go around, they'll float around, they'll attach to amino acid, then they'll go and find their way to a ribosome, and they will try to click themselves in and find the anticodon that fits on that messenger RNA. Once they find that anticodon that fits, they drop off that amino acid, disconnect. Okay. There's a really excellent video that's going to show this process in real time in the Google Classroom. It's the one that I said has the 3 sections to it: the structure of the DNA, DNA replication, and then the last piece has transcription and translation. It shows it really well, much better than me waving my hands around does. So the tRNA has a 3-base anticodon, the opposite codon to what the mRNA has, that binds to a particular amino acid. After the tRNA has dropped off its payload, found that anticodon that it fits into, dropped off that amino acid, it goes and picks up that same amino acid again. Once the amino acids have arrived at the ribosome via the tRNA, the ribosome binds those amino acids together. It creates a little peptide bond between one amino acid and the next one. It's just going to keep building a chain. So I believe I've got a diagram coming up here in a second. The last bit then is when we get to the end of that sequence, when we get to that terminator codon, we know that at that point, that is the end. The ribosome disconnects, right? The mRNA disintegrates, meaning that we've now got some free nucleotides that can be recycled and used again for either tRNA or messenger RNA. And then the whole amino acid disconnects. That string of amino acids disconnects and heads off to bend, fold, and twist into the protein that it should be. So let's have a look at what's going on here. Okay, so I'm going to try to move my video so you guys don't end up getting anything chopped off. So here is our, in green here is our ribosome. Okay, this long snaky thing over here is our messenger RNA, and it's being read, in this case, from left to right. Okay, it's being read from here in that direction as indicated by the arrows. There's a large subunit and a small subunit on the ribosome. Okay, and what it does is just kind of clomps on and reads it. I believe we're looking at a cutaway view of this. Okay, and so what's happening is we're reading in threes here. So you'll notice that down here, this is a codon, that's a codon, there's a codon, there's a codon, there's a codon, and so on and so on. And what's happening is all of these little transfer RNA molecules are coming in, and you'll notice they have a piece that would fit onto these, and then they've got an amino acid attached to them. So as they come in, here's a tRNA, it finds a place where it fits. Once it finds a spot that fits, it drops that little amino acid off, and it gets added to that growing chain of amino acids. Okay, that growing chain is basically a newborn protein. What you can see is because of the chemical structure of those amino acids, they are this particular protein is already starting to twist a little bit into the shape that it will be ultimately. It may take a little while and a much longer molecule to come out before it bends, folds, and twists into the protein. What happens is once this tRNA here has dropped off its amino acid, which would be this one right here, it disconnects and floats off to go find another amino acid. Then the next guy plunks in, drops off its amino acid, it will disconnect, and this one's going to come in, find its anticodon, and drop off its amino acid and head off into the distance. Okay? So I hope that makes sense. That animation I've been mentioning, it will do a really good job of showing you guys what you need to know on this section. What I want to do right now is I'm going to move you through how I might ask you to work with some of this stuff. And what I'm usually going to do is present you with a sequence of DNA, and you guys are going to have to transcribe and then translate what's going on. So I'm telling you right now, you've got this particular sequence of DNA. Okay, very short sequence. It's going to have a start codon, it's going to have a stop codon on it. What I would ask you to do on a question is I might give you that sequence of DNA and say, let's transcribe it to messenger RNA, then translate it into those amino acids. And often I ask you for the tRNA sequence as well. And we'll get to all of that. Okay, so the first things first. If I can get this to click, there we go. We'll start out with the messenger RNA sequence. All I've done is broken this up so you guys can see the codons. Okay, I'll often put the spaces in there for you in the actual question as well. So TAC is our first codon, our first three nucleotide sequence. When you're transcribing, you're turning that into the complementary strand of that in messenger RNA, meaning we have to have uracil. So T would pair with A would normally pair with T, but, okay, we've got an issue here. We're in RNA, we don't have T anymore, we have U. So we put a U in. And then C pairs with G. And then A with U again, T with A, G with C. 3 oh, this is incorrect. 3 T's should have been 3 A's here. That's my bad. And then we've got an A and a T, a C and a G. Could you guys go in and let me go in and do that? That's embarrassing. I should have checked my work first. Okay, so this should have been 3 A's in a row. Okay, which means I probably have my protein or amino acid wrong. Okay, I'm going to show you one more thing over here. We get to the end and we have an A, which should be a T, which we change to U, and then a T and 2 A's. Okay, so that is our mRNA sequence for that super short protein that we're trying to code for. The next thing I would ask you to do is your amino acids, AA for amino acid. So AUG, I'm just going to bring this in and show you how to use this. So I've got AUG is my first one. So along the left here, find A. Then on the top, find U, and then find the one with G. So AUG is methionine. That is also our start or initiator codon. Okay. Our next one is UAC. You can see that over here. Our next one is UAC. So bring that back in, find U, find A, find C. That's tyrosine. Okay. Now I'm going to go correct my mistake here. Okay. We have our next one is AAA. So I'm going to go over here and I'm going to find A, and then I'm going to go down and find A, and then 3 A's is lysine. And so I'm just going to go in here with my pen, and because I'm a bit of a nerd, I'm going to turn it into white and pretend that you cannot see that G. I think it's lysine with an S, but we'll go with that. Lysine with an S. This is embarrassing, guys. Okay, there's an S there. So there's our lysine, okay? And so on and so on. So AGA, bring this back in. AGA is arginine. And then we're going to get to CAU. So C, A, and U, which is histidine. And then our last one is UAA. UAA. Hey, there's our stop. And that's the end of our amino acid sequence for that. The last thing I want to do is show you guys the last bit here, which is the tRNA sequence. So here's how the tRNA sequence works. Remember, you can do it 2 ways. You can relate it back to this, which a lot of people do. I often relate it to the DNA, but it's really up to you. You can basically copy the DNA exactly as it is, except everywhere there'd be a thymine, you would have a uracil. But really, technically, these two are the ones that work together because the tRNA has to have the anticodon to this, right? So A would pair with U, U with A, G with C. Then U, the opposite is A, the opposite is U, and then I made another mistake here. This should be a G, right? So this should be an AUG. And then AAA should be UUU. I got that right. And then A should be a T, G should be a C, A should be T, but instead we're replacing it with U's. So I hope that makes sense for you. That's an example of one of these. I have another example, and what I'm going to ask you guys to do for this other example is hit pause after each. I'll just reveal these gradually. Hit pause after each of these and see how you did. So try to work your way through it on your own. So, you've got this sequence in front of you in your notes. Try to get the mRNA sequence. Hit pause now. Okay, this is what you should have had for your mRNA sequence. If I made a mistake, catch me on it, but I'm just doing a quick look and it looks good. Now, get ready to do your amino acid sequence. Have your data booklet out to that messenger RNA codons table. Okay, hit pause now. Okay, this is what you should have had for your amino acids. You guys can, by the way, you guys can shorten these down if you want to. I just wrote them out in full, but if you're running out of room, you could do a 3 or 4 letter abbreviation. I had to shorten phenylalanine or else it would have run into here. Remember, there's an alanine and a phenylalanine, so make sure that you guys watch out for that. There's also a leucine and an isoleucine as well, so pay attention to that as well. So there's your amino acid sequence, and then hit pause now. And then here is your tRNA sequence, and that should be it. All right, so hopefully that worked well for you. If you need some help with that, track me down, let me know. So what I'm going to do now, that's the first section I wanted to go through with you. So I'm going to remind you of a couple of things at this point. There's a couple of really good videos that I want you guys to have a look at. So have a look at the protein synthesis video by the Amoeba Sisters. Take 9 minutes out of your life and watch that. It's really good. And then take another 8 minutes and have this molecular visualizations thing and give it a look. It's going to go through the DNA molecule again, so that'll be a nice review. It'll go through the DNA replication thing again, which will be a nice review. And then finally, it'll get into that transcription and translation stuff right at the end. And then please now go, if you do anything, please have a look at and work on worksheet number 1, transcription and translation. The key is in the Google Classroom under Unit C, resources, keys to worksheets. Okay, that is the end of concept C10 and the end of the first video for this week. I apologize, this was a long one. I hope that this made sense. If it didn't, please track me down online and I would be happy to help you guys out with this. I hope that you're staying safe. I hope you're staying healthy, and we'll talk very soon in concept C11.